Let's have a look of the dorsal raphe nucleus. So this nucleus is one of the raphe nuclei, and it is situated in the brainstem as the midline, and it has many subdivisions as we can show in this graph. The main function of this nucleus is to secrete serotonin, the serotonergic efferents to many brain regions, like hippocampus, limbic lobe, and amygdala. So, dorsal raphe nucleus is the largest serotonergic nucleus and provides a substantial portion of the serotonin innervation to the forebrain. This nucleus is also the target of the selective serotonin reuptake inhibitors, which is used as an antidepressant. Below are some research on this nucleus. It is known that the endogenous opioid receptors exist in dorsal raphe nucleus. It is also related in circadian cycle, the sleep-wake cycle, and some patients with problem with this nucleus might have narcolepsy, which is the tendency to sleep. This is because that raphe nuclei provide feedback to the superchiasmatic nuclei. So this nuclei is directly related to circadian rhythms. Some study shows that if you remove this region in the mouse, and the mouse will have no circadian rhythms. And this is actually a bidirectional interaction between raphe nuclei and suprachiasmatic nuclei. The SCN transmits to the raphe nuclei via the dorsal medial hypothalamic nucleus, altering serotonin levels for sleep-wake states, and the raphe nuclei will then transmit feedback to the SCN about animals' vigilance and the level of alertness. So this is a reciprocal feedback. And this region is related to depression and suicide, as from here. The main target of SSRI is the dorsal raphe nucleus. Some study shows that dorsal raphe may be decreased in size in people with depression and paradoxically an increased cell density in those who die by suicide. We can see the location from this picture that the raphe nucleus is located in the middle of the brainstem, and its serotonin pathways affect multiple regions of our brain, including the amygdala, the hippocampus, the limbic system, and the whole neocortex. It can affect our mood, our memory, sleep, and cognition. So this is a very important nuclei in our brain. And lastly, the dorsal raphe nucleus also show to be the target of ghrelin. The ghrelin is a peptide produced by gastrointestinal tract. It is the signal of hungry. So if you have many ghrelin, you will feel hungry. So when administered with ghrelin, it will act centrally on the raphe nucleus and will increase the food intake. Let's have a look at the paper. The decision to move or refrain from movement depends on the structure of the environment and the internal state of the animal. So depends on different environmental states, we will behave differently. So if there is imminent threats, it may provoke a fight or flight response, and if this threat is remote, we might enter vigilant freezing or a delayed strategic avoidance response. So basically, the first paragraph talked about that different environmental state will stimulate different behavior. The second paragraph introduced the function of serotonin. Dopamine promotes behavioral activation in vertebrates. Forebrain serotonin is more strongly associated with behavioral inhibition. Elevated serotonin promotes pausing and waiting, whereas reduced serotonin promotes impulsivity and preservative responding. So this means that if you have a low level of serotonin, you will be impulsive and you will have a tendency to act quickly without careful thought. The preservative responding means that you're doing something over and over again. That's beyond what is necessary. So as we can see from this example, the serotonin is very important for inhibit inappropriate or unnecessary behavior. Paradoxically, selective serotonin reuptake inhibitors reduce immobility in the forced swim test and tail suspension test, and as that stimulation of the prefrontal dorsal raphe nucleus projection. Selective serotonin reuptake inhibitors is a class of drug that is used to treat depression. As we know that neurons transmit information by neurotransmitters, and those neurotransmitters are picked up by the postsynaptic neurons. So those neurotransmitters will bind to those postsynaptic neurons and it will activate these postsynaptic neurons. These neurotransmitters will be uptaken by the presynaptic neuron for several reasons. The first one is to recycle, because it won't need to synthesize those neurotransmitters again. It will be just recycled and ready to use for the next time. The second one is to terminate this signal. If the neurotransmitter stay in this synapse, it will continuously stimulate the postsynaptic neuron and result in a continuous activation of the postsynaptic neuron. This neurotransmitter reuptake will terminate this signal. The third function of this uptake is to prevent toxicity. If neurotransmitters remain in the synaptic cleft for too long, it can lead to excessive stimulation of the postsynaptic neuron, which can be damaging for this postsynaptic cell. This reuptake helps clear neurotransmitters to prevent excessive excitation and potential neurotoxicity. For the patients with depression or anxiety, it is believed that there's some problem with their serotonin level. So if you use these serotonin reuptake inhibitors, it will inhibit this neurotransmitter uptake by presynaptic cells. So the previous study shows that SSRI can reduce immobility in the forced swim test and tail suspension test. They used fiber photometry to monitor the population dynamics of dorsal raphe nuclei serotonin neurons in freely behaving mice. They injected this stuff into the dorsal raphe nucleus of the third Cree mice and implanted an optical fiber over the dorsal raphe nuclei. So let's draw the brain of the mouse, the brain stem, and this is the head of the mouse. The dorsal raphe nuclei is located in the middle of the brainstem, and they used the fiber to monitor the activity of dorsal raphe serotonin neurons. This fiber is like a camera that can capture the activity of neurons located in dorsal raphe nuclei. Then they injected this stuff into these mice. So what is this stuff? The AAV 5 is the adenovirus that looks like this, and we can use this virus as a vector to integrate the DNA sequence of interest to mouse genome. So if this is the mouse cell, and this virus will infect this cell and integrate its DNA sequence into the host genome. So if this is the host genome, it will integrate its own genome to the host genome. The CAG is a promoter sequence that will promote the expression of the gene. The flex is a genetic switch system, and it has a DNA sequence called LOXP. When a gene of interest is flanked by this LOXP DNA sequence and under a recombinase called CRE, this gene in the center will be reversed. So it will be like this. This is a very useful tool and commonly used in neuroscience. So if you have a gene of interest, and if you want to selectively express this gene under certain tissue or certain brain region, you can just inject the inverted version of this gene and conditionally express the CRE enzyme in the cell that you want to express this gene. This graph, it shows the GCAMP. So in the absence of calcium ion, the green fluorescence protein chromophore is exposed to water and exists in a protonated state with minimal fluorescence intensity, shown in this graph. There is no calcium ion present. If the calcium ion is bound to the chemogene, so upon calcium ion binding, the CAM domain undergoes conformational change and tightly binds to the egg. And this third CRE mice is genetically engineered mice. The third is serotonin transporter. It is a protein that facilitates the reuptake of serotonin from the synaptic cleft back to the presynaptic neuron. The CRE is the CRE recombinase, which is an enzyme that can excise or flip a specific DNA sequence. I drew it in here, the CRE recombinase. So basically what they do in here is to selectively express the calcium indicator in the dorsal raphe nuclei so that we can monitor the activity of neurons in dorsal raphe nuclei. The first test they used is open field test, which is place the mouse in an open chamber, and this test is used to measure the activity level and anxiety of the mouse. Because this chamber is new and novel to this mouse, it will explore its surroundings. If this mouse is depressed, it might just sit in there, and if it is not depressed, it will actively moving around and exploring the environment. Just like the forced swim test, there is a camera used to monitor the activity of this mouse. But the movement onset in the open field test is associated with a robust reduction in DRN serotonin fluorescence. as we can see from these three figures. The first one shows the movement and the intensity of fluorescence. The red line shows the speed of the mouse, which is measured by centimeter per second. The black one shows the intensity of the fluorescence, which is measured by delta fluorescence divided by the baseline fluorescence level. If you do the correlation analysis between the speed and the fluorescence level, you can see that the movement is negatively correlated with the movement. So after the movement, the fluorescence level will always drop. They performed different tests. The first one is open field test we mentioned before. The second one is the reward approach task and avoidance task, as shown in this figure. So in approach task, basically you divide a chamber into two small subchambers, and there is a reward in another chamber. It can be some water or sweet. So if the mouse crosses within 8 seconds, it will receive the reward. If it failed to cross the chamber, it will receive no reward. The avoidance task is similar. It just replaces the reward with some negative experience. So in this case, they used electric shock. If the mouse failed to cross the chamber, it will shock the mouse. If this mouse managed to cross the chamber, this electric shock penalty. And we can see from the result in here, the movement is negatively associated with the fluorescence level. This three experiment shows that the activity of dorsal raphe nuclei neuron is negatively associated with the movement under low or moderate threat. They next switched this low or moderate threat to high threat with tail suspension task. So under this high threat, it shows that the intensity of fluorescence is positively correlated with the movement. And the picture is very clear here that under low threat, the activity of dorsal raphe nucleus serotonin neuron is negatively associated with the movement. But under high threat, the activity of those neurons is positively associated with the movement. One problem I think is worth mentioning here is that the author used 3 low threat tests but only 1 high threat test. I think it's better to use at least 2 high threat tests to further validate this finding. The previous experiments shows correlation between the two. They next probe the causality between the neural activity and the movement. They used optogenetics to activate serotonin neurons. The optogenetics insert these light-sensitive proteins to the membrane of the neuron. If you flash a light on those neurons, those proteins will stimulate the neuron, fire the neuron. So you can artificially fire a group of neurons by flashing light on these neurons. They used channel rhodopsin 2 as the light-sensitive protein and enhanced the yellow fluorescent protein as the control protein. We can see from the result in here that in 3 experimental groups, the activation of dorsal raphe nuclei, serotonin neurons, will decrease the speed of the mice, compared with the control group, and this result is consistent in all three environmental conditions where low threat is present. But on the high threat, the result is inversed, so that the activation of these neurons can increase the movement of the mouse. And they did the same experiment, but this time they tested the GABA neurons in dorsal raphe nuclei. And this time they find that the activity of the mice decreased upon movement to cross the chamber in the approach task, but increased upon movement to cross the chamber in the avoidance task. So we can see from the figure C, is the approach task, and the figure H is the avoidance test. So for GABA neurons, it shows different firing pattern compared with serotonin neurons. In positive environment, where reward is present, the activity of GABA neuron is negatively correlated with the movement. But on the negative environment, where penalty or threat is present, the activity of GABA neuron is. This report shows that there is a movement disorders clinic was stricken by a major earthquake, and there are many Parkinson disease patients living in this clinic. There's a 14 stage 3 to 5 patients in here. So basically, the patients with Parkinson disease in stage 3 to 5 cannot move. But during this earthquake, those patients show a sudden and brief period of mobility following emotional or physical stress, which is often caused by unique life-threatening situations in patients with advanced PD, even though those patients cannot move under normal conditions, they actually movable. Another example they showed is dopamine-depleted akinetic rats attempt to escape in deep water. People believe that dopamine promotes movement. If you deplete dopamine, the rats will go akinetic. But in deep water, life-threatening situation, those rats can actually move and escape this environment, which also suggests that alternative motor regulation in fight or flight situations. And they also showed other examples of this alternative neural regulation under different environmental states. Lastly, they concluded that our experiments probed the neuronal dynamics and functional roles of two distinct dorsal raphe nuclei cell types in movement regulation, and they find that urgent escape conditions switch dorsal raphe nuclei serotonin neurons from suppression of movement to facilitation, and that dorsal raphe nuclei GABA neurons selectively facilitate movement in environments with negative valence, consistent with the neurodynamics that we observed in both cell types. These results reveal a role for dorsal raphe nuclei circuits in rapid environment-specific behavioral regulation. I think this paper is neatly presented and shows some interesting results. But one downside is that, in my opinion, the only one downside is that they used 3 low threat tests, but only 1 high threat test. If they can add another high threat test, it would be perfect. So thanks for hearing. Hope you learned something new, and see you next time.